Hello and welcome, everyone. This is the third and final podcast for Chapter 13, Day 21. In this podcast, we're going to talk about some of the specific steps of meiosis as well as the big picture of meiosis. Why is it that we have meiosis? We'll also link it into Objective 4 because it's really hard to talk about meiosis without considering how genetic diversity is created by meiosis and this evolutionary significance. Okay, let's go ahead and get started. Let's go ahead and begin our discussion of meiosis. Meiosis is one of my favorite topics. It really helps explain much of biology as well as a foundation to understanding evolution. However, I must say when I was a student, I really disliked the topic of meiosis, and probably because it seemed to come up in so many different classes, and I believe I just my eyes probably just glazed over when the instructor said meiosis. So what we're going to talk about for this course as far as meiosis is just kind of the bare essentials of meiosis, why it's important. We'll have some detail, but not as much detail as you're going to get as you move up. Meiosis will appear again, certainly in genetics and maybe in cell biology as you move forward. So let's write some features of meiosis. The first that you should be aware of is that meiosis occurs in the gonads. And of course that means your testes or ovaries, depending on genders. Remember, mitosis occurs in all of our cells, but meiosis, the specialized cell division, only occurs in the gonads. The second thing that you should know, meiosis is different than mitosis in the fact that we go from a diploid cell to typically 4 haploid cells. And remember, we covered diploid and haploid before, but diploid means having 2 copies of each chromosome, and haploid means only having 1 copy. We often write diploid or symbolize diploid by putting 2 in and haploid as just an N. So 1 copy versus 2 copies. The third point here is that meiosis generates diversity, generates genetic diversity. As we move through our discussion of meiosis, I'll stop and point out the places of meiosis where genetic diversity is generated. Meiosis, we can simply define meiosis as saying it's one round of duplication or replication. I'm going to write replication. One round of replication plus 2 rounds of segregation. So to kind of illustrate that, let's think about our cell cycle. We have G1 with S phase here, G2, and then we enter into mitosis or meiosis, but in this case it's going to be meiosis. This 1 round of replication occurs in S phase, shown here, and then the 2 rounds of segregation occurs in M phase. Remember, with mitosis, there's only 1 round of segregation, and that's why we end up with identical cells. But in meiosis, we have 2 rounds of segregation, and that's how we end up going from 1 diploid cell to 4 haploid cells. These 2 rounds of segregation can be broken into meiosis 1 and meiosis 2. They're the same 4 stages we had in mitosis, except we are going to break them down by calling the first set prophase 1, metaphase 1, anaphase 1, and telophase 1. And then meiosis 2 just redoes this again. So we call it prophase 2. I'm just going to write 2 here. Metaphase 2, anaphase 2, followed by telophase 2. We're going to focus on prophase 1, metaphase 1, and that kind of links into anaphase 1, the point we want to make about that. And then we're also going to focus on metaphase on the next slide. You'll see on the next slide that meiosis 1, its goal, one of the things it does that's really important, is that it segregates homologous chromosomes. So I'm just going to write SEG, homologous chromosomes. I'm going to abbreviate this just because of my space constraints here. So it segregates homologous chromosomes. Meiosis II does something a little different. It segregates sister chromatids. Okay, so let's begin our discussion of meiosis. Meiosis is a very complicated subject, and it has many layers of complexity to it. We're going to try to stay closer to the surface, just illustrating the things you need to know for meiosis. And as you move through the curriculum in the department here, you will be exposed to extra things about meiosis. I'm going to draw in this corner here the areas that we're going to really focus on. The first is going to be prophase 1, so I'm just going to put P1. Metaphase 1, I'm going to put / anaphase 1, because the part about metaphase 1 that's important is related to anaphase 1. And then metaphase 2. These are the 3 parts that we're going to focus in on the most. We'll talk about some of the other parts, but these are the critical parts. As we move through the stages of meiosis, I'm going to highlight the areas that generate genetic diversity. So let's begin with our cell cycle. We have G1, remember, then we have S phase, and we have G2, and we have M. Remember, in G1, before the chromosomes are duplicated, they might look like something like this. Remember, in humans, we have 23 pairs of chromosomes, but for simplicity, I'm only going to draw 2 pairs here. So 2 large red chromosomes and 2 small red chromosomes. Green chromosomes. Remember, these red ones are called homologous chromosomes to each other. These green ones are called homologous chromosomes to each other. Comparing the red to the green, that would be called non-homologous chromosomes. As they're copied, these cells, each of these are duplicated. The chromosomes are duplicated during replication at S phase. And then that enters meiosis. So what I'm going to do here is I'm going to draw a cell in prophase 1. Remember, this cell here went through S phase. It was copied. The chromosomes were copied. It goes through G2, and now it enters meiosis. The very first stage of meiosis is prophase 1. And so let's draw what the chromosomes look like here. And remember, they're going to be copied because they've already gone through S phase. So we have this chromatid, and then it's copied chromatid here. And same thing with the other red chromosome. And the green ones do the same. So let's draw the green ones too. During prophase 1, we have our first instance where genetic diversity is increased. So let's draw number 1 here. And what this is called crossing over. Crossing over occurs during prophase 1, and it occurs between homologous pairs of chromosomes right here. And what happens here is we have an exchange of genetic material of DNA between the homologous chromosomes. And because we're swapping out the DNA, this can generate mutations, and it can change the presentation of these genes on each strand. So this physical exchange of DNA during crossing over increases genetic diversity. So I'm going to draw an up arrow and write genetic diversity. Up arrow just means increase. So an increase in genetic diversity. In fact, this is probably one of the most important ways genetic diversity is increased that fuels evolution. After prophase 1, we're going to enter metaphase 1. In this part here, I'm also going to talk a little bit about anaphase 1. So let's go and write that down too. Anaphase 1 comes right after metaphase 1. Okay, so I'm going to draw the cell here. And remember, just like in mitosis, there's this imaginary line that comes down here, this center line here. And on the last slide, if you recall, I said one of the most important things that happens during meiosis 1 is that we segregate the homologous chromosomes from each other. So we want to take this red chromosome here and put it in one new cell. And we want to take this other one and place it into another cell. Same thing with the green ones, and same thing with all the chromosomes. And to accomplish this, what happens is that the red chromosomes, homologous chromosomes, line up on either side of this metaphase plate line here, as well as the green chromosomes. Now, when the centrosomes over here send out their microtubules, the spindle fibers, they will attach to each chromosome, like so, and pull it apart. They will pull it this way. And on this side, it's going to pull it this way into this half of the cell, and these into the other half of the cell. Now, this also increases genetic diversity. And so how does it do that? It does that because the way that the chromosomes line up along this center line here is completely random. For instance, this red chromosome that's on the right now could have ended up on the left, and vice versa. And the green chromosomes, the same holds true. How they line up on either side of this center line is completely random. And because of that, we get differing assortment of the genes. And so what we're going to call this is random assortment. And this is our second instance of increasing genetic diversity. Now we're going to proceed after metaphase 1 and anaphase 1. Remember, anaphase 1 is just a pulling of the chromosomes. And at the end of telophase 1, let's go into write telophase 1 here, we end up with two cells. One cell that contains one of these homologous chromosomes, like this one here, and one of the green ones. So it's going to contain this red one, and then this other cell is going to contain the other red one. Now we're at the end of meiosis 1, and we want to enter into meiosis 2. We're not going to spend much time on meiosis 2, but we want to focus in on metaphase 2. But let's just mention a little bit about what happens next. After meiosis 1, at the end of telophase 1, each of these cells, we're only going to follow one of them now, but each of these cells enters meiosis 2. It will start with prophase 1. There's no crossing, prophase 2. There's no crossing over that occurs in prophase 2. That's only in prophase 1. And then we enter into metaphase 2. So I want to talk a little bit about metaphase 2. So let's write metaphase 2. I'm going to draw a larger cell here. We have our center line here. If you remember on the previous slide, I said that an important thing that happens during meiosis 2 is that the sister chromatids separate. So if you look over here, at the end of telophase 1, we have a red sister chromatid on one side, and the other red one is the other sister chromatid. Same thing with the green. So to accomplish this, what happens is that one of the sister chromatids will be on this half of the center line, and the other will be on the other half. The same is true for the green ones. I should have drawn that closer, but they are still connected at their centromeres. Bad drawing on my part. But the key is that each of the sister chromatids are on the opposite side of this center line. And that's important because when our centrosomes here send out their spindle pulls, and then during anaphase 2, they'll pull these sister chromatids apart. So then at the end of meiosis 2, what we end up with, so after anaphase 2 and telophase 2, what we end up with are cells like this. I'm just going to draw two of them here. Each one will contain the large red chromosome. Now just one copy of it though, because now we generated a haploid cell. Okay, so remember, as we enter into prophase 1, these are still diploid cells. At the end of meiosis 2, we have haploid cells. And these will be the gametes, right? These are our gametes. And this will bring us to our third point where genetic diversity is increased. So write number 3 here. And I'm going to write random fertilization. And this increases genetic diversity. We say this increases genetic diversity because which gametes are chosen is completely random. And then when they fertilize either the egg or the sperm, depending on what we're starting here with, but let's pretend these are sperm, okay? So which sperm is chosen to fertilize the egg is completely random, and which egg is released at that particular month is random as well. Okay, so that ends the main concepts that we're going to talk about as far as meiosis. I want to close with three kind of take-home messages from this chapter. So here are the three take-home points from this chapter. These are the kind of the big picture points of the chapter. The first is that sexual reproduction generates genetic diversity, whereas asexual reproduction does not generate genetic diversity. The second point is when we're thinking about this genetic diversity, I misspelled it, but there is an I here I put above it. We know that this genetic diversity is generated during meiosis, these two steps here crossing over in chromosome segregation, and during fertilization. The last take home point, and this is a pretty big one, is that meiosis helps us understand, it helps us explain some of the central parts of evolution. Well, that's all we have for this chapter. If you have any questions, please make sure you come see us, and we'll see you in class. Bye.